check on the renal genitourinary diseases, and this is what I've just said is a course offered in the course of Kenya Registered Community Health Nursing students. So under it, we're going to see on the structure of the kidney. For basic analogy, you should be in position to diagrammatically draw a kidney, give it structural orientation, give our location of the kidney on the aperture, the gross anatomy, where they are located, whether it is retroperitoneal cavity, and so on. So you know the internal and external structure of a kidney on basis of from the urethra, the renal capsule, the renal papilla, the major calyx, all those minor calyx, connection to minor calyx, the renal pyramid, and the sinuses. Again, away from that, you're supposed also to be in position to check on the functional anatomy of the kidneys and the renal genitourinary structure. Away from that, you should be able to be in a position to directly point at the location of the kidney and the patient's abdomen. And you also need to directly give an instruction by position of the urethra, bladder, and the urethra. So away from that one, you also need to be devolved into now the functional anatomy and talk about the parts of the nephron. Discuss nephron, because nephron is what makes around 1.3 million of the kidney, a human kidney. So on the part of the nephron, you need to know the afferent, efferent arterioles, glomerular complex, ascending and descending tubules, loop of venal, and collecting duct as well. Yeah, you should also know about the two cell layers separating blood in the capillaries and the filter in the Bowman's capsules. And these layers are separated by basal lamina, and you should know about the stellate cells, which are mesangial cells, which are located between the basal lamina and the endothelium. These cells are contracted, and their role is to regulate glomerular filtration. They also secrete various substances, take up immune complex, and production of glomerular diseases. Function of the kidneys: regulation of water, electrolytes, that is one. Body fluid osmolarity and electrolytes, concentration to acid-base balance, excretion of metabolic waste, regulation of arterial pressure, secretion of hormones as well. And again, nephron, you should know what is on the video, what you're watching. If you are using the YouTube, you see on the nephron, you need to be in a position to label the proximal convoluted tubule, the neuronal corpuscles. Yeah, check on the descending and ascending loop of venal, on the entire renal tube up to the collecting system. You should be able to draw it and label it well. The nephron and the urine formation, you should also be able to explain and describe it for you to get a theoretical marks. The sphincter muscles in the blood as well, where they are located. And again on the urethra, you check about, in male is about 20 centimeters, that is 8 inches long. And the female is 3 to 4 centimeters, that is 1.5 inches long. Talk about the short length in female being one or more urinary tract infection predisposing factor than males, because the shorter the length of the urethra, therefore the inner bladder and all the triangle, the angle, that is next to the bladder is highly exposed due to this distance to any urinary tract infections. The diagrammatical presentation is what is so labeled, and we have a region called tragon. In the tragon, this region here, this tragon is very, very prone to infection, because at most there are three outlets, an inlet. There are two inlets, one outlet, and those places are forever moist. They are moist, so by that they can habitate infection like bacteria causing infection, and so that region is normally prone to the infection of the bladder. Now the division of urethra as well, you really need to know. We have segmentation or division of the urethra. Yeah, we have the spongy urethra, right? Membranous urethra. Yeah, there are actually three: membranous urethra, and we have the first part of the urethra. So away from that one, let's proceed. Let us now start on what you're supposed to cover today, that is investigative procedures. The investigative procedures, these are very key procedures that are necessitated during any condition related to genitourinary diseases. So if a patient presents with any of the conditions, you either way need to confirm the diagnosis, and the diagnosis is basically confirmed by a well and coordinated investigations. So that is what we call the investigative procedure. So in front of you there are a list of several. We have intravenous urography, or what we can also call the intravenous pylography. You will see most probably patient present with back pain, some present with very discolored urine, and again, for us to roll in, maybe we need to have a very contrastful image that can give us a well denoted, or maybe a picture. If it is CT scan or X-ray, that will help us to interpret, So we go for intravenous urography. Intravenous urography, we'll discuss it ahead. That is very key thing. You'll find we are injecting a given dye or iodinated substance through the intravenous into the blood, and that is now enabling us to have a clear detailed circulation in the kidney. Therefore, the image will be showing whether there is blood in the urine, right? So that is us. We also have urethography. We have cystogram, cystoscopy, urine, and we check on the urinalysis, renal biopsy scan, and renal function test, RFT. So these ones are what will fall on this today's class, and we'll get to understand. So let's start with the first one, that is intravenous urography. The kidneys do not show up well on an ordinary X-ray of the abdomen. This is a test that uses X-rays and a special dye to help us assess the kidneys, ureters, bladders, and the urethra. So at most, it's very interesting when we use X-ray. We normally figure on the fracture and orthopedics and all the investigations. But here, X-ray can give us the angiogram. It can give us a nice picture of the circulation as well as some most if we use the dye or staining techniques. So when we take a contrast dye, the one which is mostly is called the urographin dye. The contrast dye is iodinated, so is injected into a vein, and then the dye travels on the bloodstream, concentrates in the kidneys, and is passed out in the ureters with urine made in by the kidneys. There is greater details of the internal structure because now the dye is going to be shown in the image. It is also possible to assess roughly how well each kidney is working by the same technique. Another example is urethography. Urethography, now the radiographic study of the urethra using iodinated contrast medium, is generally carried out in males. Urethography is usually carried out to show the cause of poor urinary flow, thought to be caused by narrowing or stretch of the urethra. When you try to urinate and then the projectile force of the urine does not go far, instead it just drops next to your leg, there's a problem with your flow of urine. So when you take urethrogram, we carry out to show the cause of poor urine can be due to narrowing of the course of the urethra, and so it has to be rolled in and check whether it is a biopsy and required, or maybe there is a tumor or a polyp or anything which can be blocking or resulting to that constriction. The most common cause of narrowing of the urethra in men is benign enlargement of the prostate gland. So that is the benign prostatic hyperplasia. So cystogram is another one. Cystogram gives us cystography, which is a diagnostic procedure that uses X-ray as well to examine the urinary bladder, because it has something to do with the urinary bladder. So the contrast, which is a dye, is injected in the blood as well, may indicate how well the bladder empties. So you need to be, for you to conduct this procedure, you should also understand the anatomy, the location of the bladder, and all the contraindication and indication of this, and then you need to have all the facilitated equipment or material required for the procedure readily available. So this one may indicate how well the bladder empties during urination and whether the urine backs up into the kidney. So this one takes all normal good time duration for us to have a conclusion of the results, because we have to wait for the bladder to be full. We again check on during the urination procedure and see how the bladder does the role. Cystoscope is another one. This is a procedure to see the inside of the bladder and urethra using a telescope. So cystoscope, a long thin instrument with an eyepiece. Eyepiece is like a camera, the external end, and a tiny lens at the light on the hand. And this light helps us not illuminate inside the bladder. So it's inserted in the bladder via the urethra, or we can even form a cystomy, a direct hole, which now will lead us directly to the bladder. So the small lens magnifies the inner lining of the urethra and the bladder, allowing clear visualization. And you have to be very keen, and you also need to know what you are looking for. So it's good you be familiarized with the context of the human atlas anatomy. Urine analysis or urinalysis is very useful test in diagnosis of us and screening for many diseases and conditions, and even pregnancy is one of them. So resulting of urinalysis may help in diagnosing urinary tract infection, that is UTIs, kidney diseases, and diabetes as well. So renal biopsy is another one. and we shall check also on this. So in renal biopsy and scan, renal biopsy is just a procedure that involves taking a small sample of tissue. Biopsy means you take a live tissue from any part of the body. So it takes a small sample of the tissue from the kidney for laboratory analysis. So the biopsy is taken to one, diagnose suspected kidney problem. Any sehemu unakata kidogo unapeleka kwa lab. You can use the microscope to evaluate. You can use anything. So it's important to determine the severity of a kidney disease, monitor treatment for the kidney disease, and so on. So there are two different types of ways to perform a renal biopsy. The most common type of renal biopsy is called the percutaneous biopsy. Percutaneous biopsy, so that is or a renal needle biopsy. So one, just take on the top kidneys part of what is the targeted part of the renal, or we take a renal needle biopsy, that is now we aspire the innermost tissue by negative suction pressure. An ultrasound or computed tomography scan can be used to direct the needle to specific area of the kidney. And then we have an open biopsy or surgical biopsy. This allows the physician to look at the kidney and determine from which area the tissue sample should be taken. So the next one is renal function test, that is RFT, which is very poignant and important, and you should know about it. So the kidney function tests are common lab tests used to evaluate how well the kidneys are working, such as the tests include that is now the RFT, or the kidney function test, or the renal function test. It includes burn, right, blood urea and nitrogen concentration, that is referred to as burn. We have creatinine blood and creatinine clearance and creatinine urine. So if you are told to explain the component of RFT, I've said we have the burn, the creatinine blood, the creatinine clearance, the creatinine urine. And that is up to there. We call the end of our today's topic on investigative procedure. For more of detail, you can again inquire and check on further to analyze the same. So the next, we are going to check on the conditions. That is our next lesson, maybe on the next week. Thank you and have a nice time.